Welcome, online listeners and viewers, and thank you for joining this Science AAAS webinar, Discovering Antibodies in Months, New Advances in HT-SPR, Single-Cell BCR-seq and RNA-seq. My name is Jackie Oberst, and I'm the Assistant Editor for Custom Publishing here at Science. I'll be moderating today's webinar. Joining us today as guest speakers are Dr. Derek Croote from IGeneX, San Francisco, California, and Dr. Lucy Liu from Alloy Therapeutics, Waltham, Massachusetts, and whom we will learn more about shortly. Before we get started, I'd like to orient our online viewers to what's currently being seen. At the top right of your screen, you'll find a photo of today's speakers and a View Bio link, which you can click on to read more details about their background and research. To the right, you'll also see the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we will have a short Q&A session, during which we will answer some of the questions submitted by our live online viewers. So start thinking about some questions now and submit them at any time by clicking the Ask a Question tab, also on the right, typing the question into the message box, and then clicking Submit. You can also log in to your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the bottom left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Croote for sponsoring today's webinar. And now let's begin. Although it is highly rewarding to create medicines, particularly therapeutic antibodies, doing so comes with several risks. It is expensive, time-consuming, and often has a high failure rate. As such, any technique that can minimize costs and time while also increasing success is considered the holy grail in biotechnology. Coupled with advances in liquid handling, bioinformatics, and antibody expression and characterization, high-throughput surface plasmon resonance, or HT-SPR, and single-cell B-cell receptor sequencing, BCR-seq, are driving the unprecedented scale, scope, and speed of biologics development. We will discuss antibody discovery by immunizing transgenic mice engineered to produce antibodies with human variable region heavy and light chains, and by using high-throughput B-cell selection employing single-cell sorting, beacon, or Nanovial Technology. These workflows maintain native pairings and output a diverse final set of binders encompassing a wide variety of clonotypes and heavy and light chain genes. HT-SPR enables the evaluation of the largest of antigen-antibody interactions quickly and cost-effectively. It can also run parallel kinetic, affinity, and epitope specificity studies from the very start of the drug discovery process. scRNA-seq is a versatile tool for understanding cell types, cell states, and developmental trajectories in a diverse range of tissues and organisms. Increasingly, it is being harnessed to understand adaptive immune responses and facilitate a new paradigm of drug discovery. Without further ado, I'm very pleased to announce the first speaker to you now, Dr. Derek Croote from IGeneX, San Francisco, California. As a co-founder and CTO of IGeneX, Dr. Croote is developing single-cell RNA sequencing technologies that advance our understanding of immunoglobulin E biology and provide insights into the molecular interactions causing allergic diseases. His previous research in the laboratory of Stephen Quake at Stanford University focused on applying mass spectrometry, single-cell transcriptomics, and next-generation sequencing and bioinformatics to better understand a diverse set of diseases and immunological perturbations, including food allergies, flu vaccination, dengue infection, and glioblastoma. Thank you for joining us today, Dr. Croote.
Thank you for the kind introduction, Jackie. As mentioned, I'm Derek Croote, co-founder and CTO of IGeneX, an early-stage biotechnology company developing biologics to treat allergic diseases like food allergy. Today we'll be talking about three things. First, I'll provide an overview of single-cell RNA sequencing. I'll then touch briefly on the process of antibody cloning and expression. Lastly, I'll describe high-throughput affinity and epitope binning using SPR and how we gain a comprehensive understanding of antibodies through the integration of these multitude of data. My talk today will be followed by Lucy Liu of Alloy Therapeutics, and she'll provide additional insight into these and other topics. Before I begin, I'll say everything I mentioned today is work we do at IGeneX. Our process begins with blood samples of individuals with severe food allergies, in which we use fluorescent activated cell sorting to isolate extremely rare IgE-producing B cells. We then perform single-cell RNA sequencing to recover the full-length, paired heavy and light chain sequences that comprise monoclonal IgE antibodies. Lastly, we re-engineer these IgE antibodies that cause allergic reactions in allergic individuals into IgG4-based therapeutics that can block the allergic cascade. We'll start today with a fundamental question: why study single B cells in the context of antibody discovery? The answer lies in the structure of antibodies as heterodimers comprised of 2 identical heavy chains and 2 identical light chains. Importantly, these 2 chains are encoded by distinct genes on separate chromosomes, and their pairing is what dictates specificity and function. Consequently, both methods that lyse cells and lose this pairing are incompatible with recovery of antibody sequences, as illustrated by the cartoon on the right. Now, we've talked about why study single cells, but why use single-cell RNA sequencing specifically? I argue that with single-cell RNA sequencing, you gain rich insight into not only the antibody, but also the B cell that's producing it. So from single-cell RNA sequencing, you can get sequence and therefore specificity. You get gene expression, which gives you information on cell type, cell state, and developmental trajectory. You also get information on gene splicing and variant calling with some protocols, and with others, you can also get information on cell surface expression, for example, protein markers on the surface and fluorescent activated cell sorting. On this slide, I'm presenting just an overview of what single-cell RNA sequencing looks like. First, cells are isolated, and we'll talk about ways in which they can be isolated. The next step is cells are lysed, and the mRNA in those cells are reverse transcribed into cDNA. That cDNA is then amplified, and then in a process known as library preparation, barcoded in order to be compatible for next-generation sequencing. On next-generation sequencing instruments, typically Illumina machines, this could be NextSeq or NovaSeq, these cDNA libraries are sequenced, the cDNA sequencing reads are demultiplexed, and that data is analyzed, and we'll talk about a number of ways in which that analysis can occur. There are several methods of isolating single B cells. Early on, microfluidics was dominant, and this is a method that gives you really high sensitivity in terms of recovery of specific genes of interest, but it suffered from lower throughput. Next, in terms of higher throughput, is fluorescent activated cell sorting, where individual B cells are sorted into individual wells of a 96 or 384 well plate. This method suffers a little bit from sensitivity compared to some microfluidic approaches, but you gain, most importantly, the ability to sort cells based on various parameters. And in this way, you're able to positively and negatively select populations of interest and those that are not of interest. In this way, you can use fluorescent activated cell sorting to isolate extremely rare cell populations, and this is exactly what we do at IGeneX. We're after extremely rare IgE-producing B cells, and therefore need to enrich for this rare population and deplete other B cells that are less of interest for us, like IgG or IgA. Lastly, in terms of the highest throughput approaches, you have droplets or microwells in which cells are encapsulated or physically isolated into wells. This has really great throughput. You can get sequences for thousands, if not tens or hundreds of thousands of B cells. You do have some trade-offs, though, in terms of sensitivity or in terms of your ability to understand information about gene splicing or variant calling. But an advantage here is that there's lower expertise required, and a number of CROs even offer this as a service for those not ready to make an investment in the expertise or the capital required to bring single-cell RNA sequencing in-house. In terms of the molecular biology involved, I can just caution in that there are now dozens, if not hundreds, of single-cell RNA sequencing protocols, but not all are actually compatible with recovering monoclonal antibody sequences. And the reason for that is due to the way in which the mRNA is reverse transcribed and the cDNA is processed. So protocols that are compatible for antibodies involve those that produce quote-unquote full-length cDNA through processes such as tegmentation. And some of these include SmartSeq2 or 10x Genomics VDJ product. Protocols that won't work include those that are focused more on 3' end counting, where the focus is on gene expression, and you don't recover the full-length sequence needed to bioinformatically reassemble into a complete contiguous sequence. How are those sequences actually reassembled? So as a starting point, remember that next-gen sequencing on Illumina instruments produces paired end reads that are quite short, so 2 by 150, or perhaps on the MiSeq up to 2 by 300. But that still is shorter than the contiguous sequence that comprise a heavier light chain. And so what's needed here is a process known as assembly. This is done bioinformatically, and there are a number of software packages available that allow you to do this. But effectively what happens is there are overlaps in the nucleotides of various read fragments that can be stitched together to produce this full-length contiguous sequence. And that's what I'm illustrating here on the right in the various colors. You have kind of a purple V gene, a blue D gene, red J gene, and green constant region, and fragments from each of those distinct regions of the contiguous sequence are stitched together based on their overlaps. In terms of what we can do with antibody sequences, one of the really interesting things that I find very powerful is looking at clonality of single B cells. This is a process in which you take the sequences and you group them by some common set of criteria. This is often the cells that are sequences that have the same V gene, same J gene, have the same CDR3 length, and have CDR3 sequences within some percent similarity. And this can vary based on stringency required in terms of determining ancestry. But effectively, you're grouping cells that are related by the similarity of the antibody they're producing. And this is really powerful in terms of a number of interesting examples. One is detecting immune perturbations. So for example, recent infection. Another is looking at antigen-specific antibodies that are blasting based on some sort of immunization or immune response. I studied flu vaccination and can say there was a large plasmablast population that emerged between day 7 and day 21 after flu vaccination. And the cloning and expression of these antibodies from dominant clones at these time points produced antibodies that were specific to influenza antigens. Another useful approach or another useful example of clonality is characterizing B-cell affinity maturation, in which you can build phylogenetic trees that basically map over time in individuals, if you're able to get repeated samples, the trajectory of the immune response and how the antibodies have affinity matured against a given antigen. Lastly, what you can do with clonality is understand convergent evolution. And this is something we do a lot at IGeneX, and here's just one example. This is a really striking example where 7 totally unrelated individuals have recombined or produced nearly identical antibodies that target an immunodominant allergen in peanut known as RAH2. In other words, individuals with peanut allergy are basically producing the same antibodies against the same antigens that causes disease. And so on the lower left, you can see that a lot of the somatic hypermutation of these antibodies is, as we expect, clustered in the complementarity determining regions, or CDRs, based on the high level of diversity at those residues. On the right, you could see a really interesting phenomenon whereby if you cluster the sequences by their similarity, their Levenshtein edit distance, you can pull out from this data the individualistic affinity maturation process that's occurred despite similar recombinations in these individuals. So you could see on the side and at the top, the cluster map shows colors that are grouped together. These colors represent the individuals of origin for these antibodies, and the fact that the colors are grouped together indicates that despite individuals recombining similar antibodies, it's an individual affinity maturation process. Not only can we understand a lot of information about the sequences of antibodies, for example, somatic hypermutation, somatic hypermutation, clonality, replacement mutation, silent mutation, but we can also overlay information about the gene expression of these cells in order to build a deeper understanding about the origins of the B cells from which these antibodies are derived. So I'm presenting just two examples here of two different disease states. The first on the left is the UMAP dimensionality reduction prediction from the Tabula Sapiens Consortium. This is a really kind of monumental effort by a number of groups in the single-cell RNA sequencing field and led by the CV BioHub in California, in which individuals donated organs effectively, and groups associated with the BioHub, Stanford, UCSF, and Berkeley dissected those tissues, performed single-cell RNA sequencing in order to understand cell type and cell state in all tissues of the human body. And so I've just subsetted that in this example here. All the data is publicly available online, by the way, to just 264,000 immune cells, and pointed out how you can look at the clustering of these cells to identify cell state and B cell subtype. So on the left, you can see clusters of naive and memory B cells, and on the right, I'm pointing out a cluster of plasma cells that originate from the bone marrow of several of these healthy donors. On the right is a different example. Rather than from a healthy individual, this is single-cell RNA sequencing and a UMAP projection for diffuse B-cell lymphoma. And this is an example where you can overlay information about the malignancy or B-cell subtype as compared to healthy infiltrating cells, and better understand disease state across individuals and within an individual over time. So there's really power in integrating multiple orthogonal data here, not just on the antibody, but on the B cell of origin. I'll touch briefly now on the process of going from sequence to physical antibody and how to characterize those antibodies. So we talked about single-cell RNA sequencing as a method to discover the antibody. At this stage, after sequencing, you have the paired heavy and light chain variable regions. Those variable regions can then be synthesized. They can be cloned into heavy and light chain expression vectors, and those expression vectors can be co-transfected into mammalian cells for antibody production. In the biotechnology field, these mammalian cells are typically HEC293 or most commonly CHO cells, and many of the cell lines have been optimized for high titer productivity of antibodies. After these cells are allowed to produce sufficient amounts of antibody secreted into the supernatant, you can collect that supernatant, use protein A to purify the antibodies, and then elute the antibodies such that you now have the physical protein corresponding to the sequence that you discovered through single-cell RNA sequencing. In some protocols, you might already have specificity based on how you performed your single-cell RNA sequencing and isolation process. For example, with flow cytometry, you can do antigen-specific cell sorting, or some microwell or encapsulation protocols allow the embedding of some specificity information or some readout of specificity at the single-cell RNA sequencing stage. But if the protocol you've performed doesn't have that component, the next step would be specificity screening. For us, our discovery platform, known as the SeqSifter platform, is an unbiased one in which we're discovering all IgE-producing B cells from the blood of individuals. And so at the time of sequencing and antibody expression, we actually don't know the specificity of the antibody. So we screen our antibodies against collections of antigens, in this case, food allergens and other allergens, in order to identify high affinity antibodies that bind to allergens causing allergic disease. So once we're able to confirm the specificity of their antibody, the next step in the characterization process is high throughput affinity measurement and epitope binning by SPR, which will be the next step of this talk. I've shown in this slide at the bottom some example timelines just to give some general guidelines of what people might expect this might take, but these can vary quite substantially depending on the specific protocol, the scarcity of the sample, and whether all of the infrastructure is in place to streamline these steps. Now, to talk about high throughput SPR-based affinity measurement and epitope binning. These are all data that we've collected and reports I'll present on the Cartera LSA platform. We have one in-house, and it's been really a workhorse for us in terms of identifying and determining the affinity of hundreds of antibodies against all different allergens that cause allergic disease in humans. And I'm presenting just a summary overview of that data here rather than hundreds of binding curves. For several allergens, what we're finding from allergic individuals is that the affinity maturation process that's produced IgE is one in which produced high affinity antibodies to these allergens that are driving disease. So in the case of peanut, there's a couple allergens known as RAH2 and RAH6. In the case of shrimp allergy, a dominant allergen is known as PEN-A1. This is tropomyosin, a muscle protein. And then in cat, there's an immunodominant allergen known as FeLD1, and this is a salivary protein that appears in cat dander. And in all of these cases, what we're finding from allergic individuals is that the antibodies being produced are extremely high affinity, some down into the single-digit picomolar range. So a really, I think, compelling example of using human samples as an origin for antibody discovery when that's amenable to your target of interest. I'll next walk through the process of high throughput epitope binning. This is a process in which your first antibody, the one shown in black here on the left, is functionalized or captured on the array surface. This is known as your ligand antibody. You then inject in antigen, as shown in this as a red circle here. The last step is injecting your second antibody, a soluble antigen known as the analyte. And when you do this, a few things can happen based on the epitope specificity of these antibodies. In the first example, if the ligand antibody shares the same epitope as the analyte antibody, what will happen is the ligand antibody will block the binding of the analyte antibody, and you'll get no signal increase as measured by the sensogram on the Cartera instrument. In contrast, if the antibodies bind to distinct epitopes, your ligand antibody will not block the analyte antibody, and you'll get this effect known as sandwiching, which you can see as a very stark step signal increase in your SPR sensogram. The power really comes from doing this pairwise competition across all pairs of antibodies in order to understand the number of unique epitopes on an antigen. And so here I'm presenting an example that likely many of us are familiar with at this point. This is for SARS-CoV-2, in which a consortium of individuals discovered antibodies through all different techniques or developed antibodies through all different techniques. These antibodies were measured for their affinity and also for their epitope bin determined by the Cartera LSA. And what you can see here is a result of that process, which is a cluster map of these individual antibodies in pairwise form. And so antibodies that block are shown in dark blue, antibodies that sandwich are in light blue. And from the clustering of these pairwise interactions, you can understand the number of unique epitopes on an antigen and use that to basically in a process kind of funneling, reducing the number of antibodies you're studying to select antibodies to proceed to epitope mapping. So really this is a nice step in reducing the collection of antibodies that you need to study down to a subset that bind to a unique epitope. For us, this is really useful in determining the immunodominance of a given epitope on an allergen. Not all epitopes are created equal, and for us, in terms of that's especially true for driving allergic reactivity. So we use this quite extensively in order to determine to which epitopes a majority of individuals with an allergic disease are producing antibodies against. Overall, we've talked about kind of a three-step paradigm now. Discover, in terms of single-cell RNA sequencing; engineering, basically identifying that sequence, producing that antibody. For us, we actually do a class switching step where we take IgE variable region sequences and graft those onto an IgG4 backbone. But in any case, it's the same process where you go from variable region sequence to physical protein that you then characterize. And we talked about using the Cartera LSA to measure the affinity and look at epitope binning of these antibodies. But it's really the first step in several additional that'll allow you to evaluate the functional properties of your antibody. I think now is a really exciting time in the field given the rapid advancement of all of the aforementioned technologies. We're now at a point where for each antibody we can gain comprehensive insight into not only their sequence, but the origin of the B cell from which it came, the B cell subtype, B cell state. We can tie in information about the specific organism. In our case, it's donor clinical information or diagnostic information, and then use that data paired with functional evaluation of the antibodies to really select optimal drug candidates. And Lucy will get into some of these aspects as well, but ones I didn't touch on today include developability and activity. Ultimately, we want to evaluate the functional ability of our antibodies in a given disease state to be kind of potent and active. And for developability, these are another orthogonal set of constraints that are applied to an antibody in terms of its ability to be manufactured. So does it have a high melting temperature, aggregation temperature? Is it sufficiently soluble? Can it be produced? Meaning, can CHO cells express at high titer? Is it pure in terms of monomer, glycosylation pattern, that sort of thing? And is it stable? And so these are all important facets of ultimately selection of antibodies for or as drug candidates. So I'll end here just by saying we're performing everything I mentioned at IGeneX and attempting to build an unprecedented understanding of IgE biology on our path to developing allergy therapeutics. You can visit us on our website, igenex.com. We're on LinkedIn as well as Twitter, and there's my Twitter handle. And I'll lastly add that we're in the process of raising a Series B. So if anyone is interested in developing therapeutics or biologics specifically that really fill this unmet need in food allergy, please do reach out.
Thank you, Dr. Croote. Our next speaker is Dr. Lucy Liu from Alloy Therapeutics, Waltham, Massachusetts. Dr. Liu is Senior Director and Head of Global Bioanalytics at Alloy Therapeutics, where she oversees development of comprehensive lead selection enabling data packages for partner discovery services campaigns. She previously served as Director of high-throughput characterization at encompassing Therapeutics, and prior to that as a project lead at Biogen. Dr. Liu received her Ph.D. in biochemistry from McGill University, after which she served as a postdoctoral fellow at Dana-Farber Cancer Institute and as a postdoctoral scientist at EMD Serrano's in vitro antibody technologies group. Welcome, Dr. Liu.
Thank you for the kind introduction. My name is Lucy Liu. So I'm from Alloy Therapeutics. Thanks for organizer for inviting me to this webinar. So today I'm going to talk about our workflow in terms of the antibody discovery from transgenic mouse and BCR sequencing. So in this talk, we'll cover humanized mouse model for antibody discovery, and then I'll talk about the high throughput characterization for lead clones. And some of this data will be included in the next session, which is a binder recovery by B cell selection and sequencing. So it includes B cell sorting, beacon, and Nanovial technologies. So at Alloy, we have a suite of transgenic mouse model that we use for antibody discovery. So on the left here, it shows the transgenic mouse models that we call it ATX-JK mice, that with different background with human CABA chain. So in all of this humanized mice, the antibody heavy chain and light chain variable domain are fully humanized. So in this CABA chain ATX-JK mice, it has a black 6 background, mouse background, as well as BOB-C, and we also have strains with a cross of those two strains. And we have to reduce the immune dominance, we have this V locus of heavy chain split into half. So the rest of the heavy chain variable domain and the light chain CABA chain is not affected. So we have these two strains, the proximal heavy chain and distal heavy chain. So each have half of this V locus of the heavy chain, so that to reduce this immune dominance effect for some target. And to explore this, to have this diverse structure diversity and also sequence diversity, we also generated a lambda chain transgenic mice with these black 6 mouse background. And also we have this hyperimmune transgenic mice. There are 4 strains with knockout or overexpression of certain genes to overcome some limitation to generate antibody discovery for difficult target. So today, all of these talks that we talk in this talk, we talk about the result generated from this ATX-JK mice on the left. So first, I'll show you the validation data we have for this transgenic mice. So first, we compare those mice to wild-type mice in terms of the splenocytes. So on the left, you can see these total splenocytes are comparable in the different strains of mice tested here. And we also tested for spleen B cells and T cells. They are all comparable to all of these strains that we tested here. And next, we test for plasma cells, and both spleen plasma cells and bone marrow plasma cells, and they show very comparable among those different strains. And lastly, we test for the secretion of IgG and IgM, test their concentration in the serum, and they are also very comparable, both under naive condition and also immunized condition. So we conclude that ATX-JK mice have a comparable immunophenotype to wild-type mice. So the next question is whether they represent the human antibody repertoire. So here on the top panel, it shows the frequency of V gene usage in terms of comparing these ATX-JK cross mice that shows in blue to the human data. This human data is published by Van der Heyden in 2017. So overall, you can see very good resemble of human reference data for the V gene. And also here, we have the J gene, the frequency also highly comparable of the two groups. And we also did comparison in terms of V identity, so that shows percentage of to the germline. So basically showing this somatic hypermutation, and basically the profile of these ATX-JK mice is highly comparable to human data. So then after verification, we use those transgenic mice for our antibody discovery. So before talking about antibody discovery here, I include some of this characterization method we used for the antibody discovered. So as mentioned by Croote, my co-presenter early, so he did a very nice summary of how this high-throughput SPR works. So here I'll just briefly mention that we can use it to evaluate purified antibodies in terms of kinetics affinity, epitope binding, and as well as these epitope mapping by using peptide library. So I want to draw your attention, in addition to evaluate purified antibodies, this method can also use for non-purified antibody still in conditioned medium. So by screening soup, we can determine the kinetics affinity by using this capture strategy. So we can characterize for affinity, cross-reactivity, both species cross-reactivity and family member cross-reactivity, and also can characterize for blocking for the ligand binding or blocking for these comparator antibody binding. So also we have cell binding assay to screen the antibodies in soups, and based on those data, we select a subset of antibody for to purify for the final set of his. And for the purified antibody, we have a panel of assays developed to evaluate those antibodies. For quality control, we use SEC, HPLC, and CSDS to determine the purity, and for DLS for hydrodynamic radius, and LCMS for mass ID, and also for PTM modification. For binding specificity and blocking, as mentioned in the previous slide, that we can use high-throughput SPR. In addition to that, we can use a cell binding assay on the right, so we can determine by this titration, determine EC50 specificity. We can also do ligand blocking assay on top of the cells, and those cells can be engineered cell lines overexpressing the target, or the cultured cell lines just natively express the target, and we can also primary cells as well. We also evaluate developability for potential develop the antibody as a therapeutic drug. So basically, we evaluate thermal stability with nano-DSF, turbidity. We test for cell interaction, polyspecificity, or called polyreactivity to different reagents, and also we assess the hydrophobicity by Higgs chromatography. So some of this data I'll include in later of this talk. So how we recover the binders after immunization. So this slide shows the platform we use for binder recovery. So we either use B cell selection or hybridoma fusion or phage display library. Basically, it's an immune phage library. We can also use combine those methods. So today I'm going to focus on these B cell selection on the top. So traditionally, people use the antigen baiting for memory B cells. Also, we call it B cell sorting. Here we can incorporate with the NGS sequencing to have more diverse antibody discovered. And this, you can select a big number of positive hits. But if you want to include some of antibody characterization, you may consider on the bottom of the two technology. One is Beacon and the other one is Nanovial. So I will talk about the workflows for these different technologies. So on this slide, it shows our workflow for single B cell sorting. So after, on the top left, after immunization of ATX-JK mice, the one is the immune tissues harvest. We sorted antigen-specific memory B cells, and then it's a single cell RT-PCR, and then we can automatically select the hits based on the sequence data. And after that is cloning and expression. In the middle of the bottom, it shows a high-throughput characterization. That's what I meant in the few slides early, that you can screen using the high-throughput SPR for the antibodies still in the soup. And we also do a cell binding for the soup screening as well, and then narrow down to a subset of antibody for purification. So on the left bottom, it also shows our NGS workflow. So basically, we explore the repertoire, and then based on the selected B cell sequence, we can select additional likely binders from the NGS pool. So this slide shows one example. This is target is TAA, tumor-associated antigen. So we immunize with a human antigen, human target, and here it shows the gating strategy. On left, it shows a selection of IgG positive live cells. In the middle plot, it shows these selection of B cells, basically IgG positive B cells. On the right, it shows these target binders. So we immunize with a human target and select here with a cyno target. So basically, we're looking for clones that cross-reactive to human and cyno of this target of the target. And so on the right, it shows the distribution of sorted cells from 4 different mice, and each mouse we have two immune tissues that are spleen and lymph nodes. So in total, we have 1,500 cells selected by B cell sorting. And then, so on the bottom it shows the clonotype distribution. Top panel shows the B cell from B cell sorting, and the lower panel shows NGS repertoire. So here you can see that the clonotype distribution and mirrors in these from the NGS data, this repertoire data, and based on the binders from B cell sorting, we can select additional clones from the NGS pool. So here the selected clones are purified, and here it shows isoaffinity plot to human antigen on the left and to cyno antigen on the right. So here these control antibodies are color shows in color that's our compared to antibody controls. So we can see that we generate antibody, we discovered antibody that can have tighter binding or similar affinity as or benchmark. And there's a lot of cross-reactive clones discovered. And we also did the epitope binning to show this broad epitope coverage. And this cell binding assay just showed this snapshot of one concentration that shows positive binding to the target expression cells, but not to the negative cell line. And in this slide, it shows convergence landscape to show this similarity plot. So basically, from this B cell sorting this project, we discovered 88 cyno cross-reactive clones that representing 46 unique clonotypes. So next, talk about plasma cell screening using Berkeley Light beacon. So after immunization, plasma cells are isolated based on the marker CD138, and then they are screened on beacon for binding to multiple antigens, for blocking and to like ligand blocking or compared antibody blocking, and for cell binding. And also importantly, we can include a functional screen on beacon. So such as the reporter cell line as it can be used. And then the selected cells will be exported and sequenced and cloned and for expression. So here is one example that basically this is showing the assay principle that you use this multiple well, the chip. So basically you see the cells in those wells, and the maximum you have 1 cells per well, and this well also called the pen. So in this case, on the right side, you have this plasma cells sitting in one well, and it secreted the antibodies, mouse antibody, and the beads that loaded with anti-mouse antibody on the top of this chip, on the top of this well, that the antibody can bind to those beads if the antibodies are secreted from that plasma cell. And the green dots represent the fluorescence labeled antigen. So if the secreted antibody binds to the antigen, you will see the signal increase with time. So that we call blooming. So here it shows one example that on the left there is a human antigen assay. So you can see some of these active clones that shows in the yellow on the top of this of those wells, of those pins. And on the right side is IgG assay. So you want to select clones that shows positive in both assays. And then once you export those clones and sequence them, so here we have plot of these we have a plot of this usage of the clonotype. So on the x-axis shows the usage of light chain region, and the y-axis shows the heavy chain region usage. So this is a heat map. So on the left, this heat map represents these clones selected by Beacon technology, and so blue it means there's no zero frequency, and the warmer the color is, the more the frequency is. So here it shows this clonotype distribution for the clones selected by from these plasma cells. To compare, we also plot the memory B cells for the same target from B cell sorting. So you can see the clonotype shows very differently on the two left plot. And to compare them on the right, so on the right side, it shows a combine of these two plots. Basically, if these clonotype used by both plasma cells and B cells are shown in purple, if used by plasma cells only shows in blue, and if it shows used by memory B cells only shows in red. So you can see the pairing is very different between plasma cells and the memory B cells. We also tested for CDR3 diversity. So here, every clone in the set is compared to all the other clones in the set. And this is a heat map. So blue, the dark blue means exactly same, the sequence CDR3. And then the warmer the color is, the more diverse they are. So the light blue means you need to change 3 or 4 amino acids to make the CDR3 sequence the same. And the yellow means you need to change 10 amino acids to make them the same. So you can, you know that the CDR3, the most abundance is like a 14 amino acid length. So you can see from this heat map, most the color is either yellow or red, meaning this is a highly diverse for this CDR3 sequences. We also evaluated the binding affinity. So here it shows in blue, the Beacon selected plasma cell derived clones has a higher affinity than from memory B cells on the right. So in summary, that these shows the plasma B cells, that shows the heavy chain and weak coupled chain family pairing are very different from these memory B cells. And also plasma cell derived clones shows higher affinity. So the next example case study I want to show you is a difficult target. So this is a TCR mimic project. So basically, we want to select antibodies that bind to bind to this HLA complex, which shown the cartoon is shown on the top. On the left, top left, in this box. So basically, the red dots indicate these peptide engrafted on the top of the HLA complex. So the HLA complex, if they are using the same HLA allele, the HLA part, the heavy chain and the light chain B2M, they are exactly same if they use the same HLA allele. So the only difference is this peptide engrafted on the top of the HLA complex. And this peptide is about 8 to 10 residues. And then some peptide only differed by 2 amino acids. So it's kind of hard to select specific antibodies. So that's why we have a few assays designed. So first on the left, the bright field image shows there's 1 cells per pin per well. And then lower in this on the left, it shows IgG assay. Basically, shows blooming of in the middle pane shows that it's IgG secretion plasma cell. And here, basically, it shows this. It shows the assay with the proteins. So here you have this HLA complex. The positive complex shows on the top, and the negative complex shows on the bottom. So it shows only reactive to the complex of interest. So next we show on the cell. So basically, this is a T2 cells. It either pulse on the red peptide or the control peptide, CMV peptide. So here you can only see the blooming happens with this positive peptide. So this is the from those assays, it shows that the plasma cell on the second bin on the second pin, it is the right plasma cell we're looking for. So based on this data, we So here the pie chart shows the percentage of positive clones that basically 22.8% shows specifically binding to this with the red correct positive peptide, but not to the negative peptide. And on the cells, we also about like a 22% shows to this presenting this red complex HLA complex, but not the negative control. Interestingly, when you compare these two groups of clones, they only overlap about 30%. Meaning, if you only do this protein-based assay, you will have less than half shows cell binding. And as the cell binding is more close to physiological condition, so it shows that it's very important to include this cell-based assay for the selection. So now I show you two representative clones that discovered from this project. There are actually two different projects, but actually on the left it shows the specific binding from the cell binding assay. It only binds to the cells with the target peptide, the positive peptide, but not to any of those 5 negative peptides. And also it shows tight binding to those cells, present those the HLA complex. So it's either sub-nanomolar or single-digit nanomolar binding, EC50. And also it shows concentration-dependent binding to this HLA complex in the protein on the protein level. So as mentioned at the beginning, we have a panel of assays to evaluate the antibody hits. So here it shows that the purity is showing a good purity shown by the SEC and CSDS, good thermal stability is shown by this melting temperature and TX, and good developability is shown by this polyreactivity. There's no polyreactivity. All right, I'll use the rest of the talk just briefly talk about these Nanovial-based antibody discovery workflow. So here, the first step is cell loading. So Nanovials, those are small container. It's like a bowl that. So here in the cartoon it shows in the red, but in reality they are transparent nanogel. So in this case, we load the plasma cells onto the nanovial. And here the nanovial internal it has this CD138 to help the loading of plasma cells. And then we can remove the free cells after incubation for about 1 hour. These plasma cells start secreting these antibody, and if the antibody recognizes the antigen, so the antigen is already loaded onto the inside of this nanovial. If it binds to this antigen, then it will tether to the nanovial. If not, then it will wash away. And after that is the staining part. Basically, use this fluorescence labeled anti-mouse IgG antibody, and then we can load to the sorter, sort out into this microplate for sequencing. The rest of these procedure of the steps would be the same as other procedures. So here it shows the gating strategy that you have this green gate showing the free cells. The red gate shows these empty nanovials, and the black gate shows the cell-loaded nanovials. And then we select by markers by CD138 and also by TISE, both are markers for plasma cells. So now we gate on the plasma cell loaded nanovials only. And if you have them at 4 degrees, which is on the top panel, you don't see any secretion of the mouse IgG. So they are only like about 0.4% background. But if you culture at 37 degrees, you are able to see some reactive to this anti-mouse IgG antibody. And then you can sort those nanovials for sequencing. For this specific project, we sorted out 130 plasma cells and 62 we are able to recover the sequence, and they represented 53 clonotypes. So on the right, it shows this clonotype distribution as well as the diversity of CDR3 sequence. So then we test for binding to protein. There like a 37% binding to ECD, monovalent ECD, and 40% show cell binding. And in the middle, it shows cell binding curves. Based on that, the EC50 can be determined, shows on right. So here it shows the Nanovial selected plasma cells. It has a higher tighter binding to the cells. Of course, we cannot conclude from this one project, but it shows the right trend. And then we also did the epitope binning to show that the coverage of epitope in the middle, the two clones bind to a distinct epitope. On the right, these two clones in the same bin. And interestingly, if you filter this, the cell binding result to this binning map, you can see the red bin with the two red clones. They bind to the same epitope, and apparently this epitope doesn't present on the cell surface, or it's buried on the cell surface and not accessible because both clone shows no cell binding. All the other clones shows positive cell binding, and when EC50 can be determined, they are very tight binding, so subnanomolar EC50 are determined. So in summary, I've shown you we validate our transgenic mice, ATXGK mice, and we use that for our human antibody discovery. And also I've shown you our B cell selection selection workflows, including B cell sorting and also plasma cell screen with on Beacon and with Nanovial technology. So a lot of people contribute to this work. I want to thank everybody who contributed to generating data for this project. So with that, I'd like to take any questions you may have.
Okay, many thanks to Drs. Croote and Liu for their presentations. We are now going to move to the questions submitted by our online viewers. A quick reminder to those watching us live that you can still submit your questions by clicking the Ask a Question tab on the right, typing the question into the message box, and then clicking Submit. The first question is for Dr. Croote. Dr. Croote, how would you advise evaluating if scRNA-seq and HT-SPR is right for a project?
I think over the last several years, the accessibility of single-cell RNA sequencing and high-throughput SPR has increased tremendously. And there are a number of CROs that are able to offer some of these services that allow you to pilot single-cell RNA sequencing on a given target. And I'd encourage that as probably a first step before making a large investment into the expertise and the capital equipment required to spin these programs up in-house. For high-throughput SPR, though, again, I mentioned earlier, I think the Croote LSA has been great for us, not just in affinity measurement, but epitope binning and guiding us towards optimal therapeutics analogy.
Dr. Liu, how would you use your transgenic mouse models in bispecific or multi-specific antibody discovery?
So basically, different light chain pairing is issue or difficulty for antibody production for bispecific and multispecific antibodies. So when we use antibody discovery, we can currently we're using common light chain immune phage libraries. So basically, you prepare from this immune tissues from immunized ATXGK mice. So basically, select the binders, antibodies against a different target from the same common light chain phage library. And another way we can do that is we also have a single domain phage library, single domain antibody. So basically, if you use regular IgG for one target, you can use a single domain for other targets in this multi-specific, so you don't need to worry about light chain pairing. And in addition to that, for transgenic mice models, we are also developing the single domain mouse model and as well as common light chain mouse models. So our common light chain mice will be available early in 2023, and our single domain mice will be available later in 2023.
Okay, back to Dr. Croote. When you reformat the IgE into IgG, do you silence the effector functions of IgG? As you know, IgG immune complexes can also lead to adverse events.
Great question. For the IgG subisotype, we've chosen IgG4 for a couple of reasons. One is because of the minimal effector function compared to, for example, IgG1. There's also precedent in this choice in the field of allergy. IgG4 increases by orders of magnitude over the course of immunotherapy. So nature is effectively telling us that in order to produce blocking antibodies over the course of an immunotherapy treatment, IgG4 can offer some of that protection. So we've chosen it for those two reasons. And then we also don't anticipate a lot of issues with complexes due to the fact that we expect to be in large stoichiometric excess of our antibody on board in vivo for individuals undergoing our treatment.
Okay. Dr. Liu, can you comment between the Beacon and Nanovial platforms and give the pros and cons of each?
Right. So the good thing is that you can include some of these antibody characterization by using both method. And then in terms of Beacon is well established, has been for quite a few years, so people using it. So then it's more very robust. But in terms of throughput, it's relatively low comparing to fax-based assays. And for Nanovial, it's a new technology that is, so it's sorting based, so it is higher throughput. Of course, some of the assays is under developing, like these sequencing, barcoding sequencing, and also cell-based assays is now under developing right now.
Okay, we have another question for you, Dr. Liu. What are the throughputs of the B-cell assays you talked about?
What is sorry, could you repeat that question?
Sure. What are the throughputs of the B-cell assays you talked about?
Mm-hmm. Yep. Yeah. So the throughput, like mentioned earlier, for B-cell sorting and Nanovial technology, it used the sorting method, fax method, and for selection, so it's a higher throughput. So then we can screen millions of cells in a day. For Beacon technology, is relatively lower throughput, so it's about 20,000 to 40,000 cells in a day.
Okay, Dr. Liu, again, are you able to rank the plasma cells for target binding affinity when you select them using Beacon versus Nanovial technology?
So unless you have IgG assay, like for on Beacon assay, you can also use for these stain for IgG secretion, so you have some idea of the secretion level, and then comparing to your target binding signal, you may have some idea in terms of binding affinity. But usually there are a lot of variables, so we usually don't rank them for affinity. We basically just for yes and no to select the cells, and then after that, when we express them, then we can do a more careful characterization in terms of ranking of affinity.
Okay, back to Dr. Croote. What are the advantages of SPR over BLI?
I'd say for both, you're able to get affinity of antibodies, and both have been used extensively for that purpose. I see an advantage of SPR in the Cartera instrument in that it also has integrated ability to do epitope binning. And that's really powerful for understanding more context of the antibodies you've discovered. Effectively, how many unique epitopes are there on an antigen, and what is the distribution of your antibodies to those epitopes? Those are all additional data you can get with this SPR design instrument.
Okay, Dr. Croote, how does developability of BCR-seq derived antibody look like, and how to improve it if needed?
For us, we're doing all of our isolation from allergic humans, and so we have quite a bit of data now on developability of human BCRs from allergic individuals, and overall quite good. Our antibodies generally express very well. But there are some sequence liabilities. Just because a human's producing an antibody doesn't mean it's optimized for high titer, stable expression in CHO cell lines at a CDMO, for example. So there will be some liabilities that often need to be removed in order to converge on a drug candidate that's optimized for the manufacturing process.
Dr. Liu, do you ever combine FACS with Beacon in the same workflow?
That's a good question. So I think potentially that you can for the selection, but I'm not for our current project. We are not using combine of these methods.
Okay, Dr. Croote, can you discuss the range of affinities found during your monoclonal antibody screening process?
I think those will vary quite substantially based on the source of the B cells, as well as the nature of the antigen or nature of the target itself. So it's hard to give a generalized answer. But what we've seen for human-derived antibodies, the affinities can be extremely high, especially in the cases where someone has undergone some sort of immune challenge or immune perturbation repeatedly. And that's exactly the case with food allergies. The current treatment for many individuals with food allergies is avoidance, in which you try to avoid what you're allergic to. And I can attest from personal experience having a food allergy that is ultimately unsuccessful. And so each accidental exposure is the opportunity for the immune system to undergo additional affinity maturation and produce high affinity antibodies. But high affinity antibodies is certainly not something that's limited to just human-derived antibodies, but there are lots of models and animals and strains that can produce high affinity antibodies that serve as foundations for drug candidate selection.
Okay, again, Dr. Croote, which script or program was used for BCR clonotype heat map?
So I'm going to assume this is related to the convergent evolution slide where we looked at Levenshtein edit distance of heavy and light chain variable regions in individuals who produced antibodies specific to a peanut allergen. This was basically a script I wrote that uses SciPy as well as the Python package Seaborn for actually generating the cluster map, and it has configurable options that allow you to add additional metadata to the antibodies. And in the case that I presented, individual of origin was one of those metadata that showed an individualistic process of affinity maturation.
Okay, Dr. Liu, how do you choose whether to run the Beacon or Nanovial process for a plasma B cell workflow, or do you typically run both in parallel?
Okay, that's a good question. It really depends on the target. For some target, like depends on the assay that we want to do. Like for example, if you have a reporter cell line, so it's more established in on Beacon that to include those functional assays. Of course, as I mentioned earlier, that Nanovial, particularly about science, they're also developing cell-based assay as well. So then it depends on the target. And also I want to comment on one, so it's related to an early question about like combine FACS assay and with sorting assay with these beacon assay. So it depends what you meant combine. So originally I understand it as like to evaluate the same set of the binders, but actually for different target, we actually use a different method to select the binders. So definitely for the selection, we have a lot of cases we use both beacon to select the binders, and we also use the FACS-based method to select binders as well. And as shown in one of my examples, that they can cover different pairing of this different pairing of V heavy chain and V light chain, and then shows a different more clonotype coverage.
Okay, we've come upon the end of the hour. It just remains for me to thank today's speakers, Dr. Derek Croote from iGenix, San Francisco, California, and Dr. Lucy Liu from Alloy Therapeutics, Waltham, Massachusetts. Please go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from science available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation within approximately 48 hours from now. We're interested to know your thoughts of this webinar. Send us an email to the address now up in your slide viewer, webinar@AAAS.org. Again, thank you to our speakers and to Cartera for their kind sponsorship of today's educational seminar. Goodbye.